NGF delivery to the brain is challenging. Here we present a new type of carrier that allows a safe and prolonged release of the bioactive protein.
This technique allows a simple and efficient loading of NGF into porous silicon carriers that is carried out at room temperature and without using strong organic solvents.
As the porous silicon carrier design is tunable, the carriers can serve as NGF reservoir implants for long-term release, but can also be modified to carry other factors and drugs.
For engineers and chemists, the fabrication process may be easier to perform, while for life scientists and clinicians, the cell culture procedures should be relatively straightforward to reproduce.
Begin by mounting a 1.5 by 1.5 centimeter silicon wafer with a well-characterized resistivity into a polytetrafluoroethylene etching cell, using a strip of aluminum foil as a back contact and an O-ring to seal the cell.
To fabricate reproducible porous silicon nanostructures, use silicon wafers with similar resistivity values each time.
Electrochemically etch the silicon sample in a 3:1 solution of aqueous hydrofluoric acid and ethanol for 20 seconds at a constant current density of 250 milliamps per centimeter squared. Then rinse the surface of the resulting porous silicon film with ethanol 3 times before drying under a nitrogen stream. When the sample is dry, thermally oxidize the freshly etched porous silicon sample in a tube furnace at 800 degrees Celsius for 1 hour in ambient air to form an